Hey, so circling in, I know it was Monday, but congratulations on the exam. Looked good again. I think we are still beating the 8 a.m. class, which is always my goal. I didn't check on this one, but you beat him on the first one, and I'm feeling good about this one. So that's good. Okay, so what's going to happen for this unit is we're going to finish up with our respiration stuff. So the exam left off talking about partial pressures. So on this next exam, you'll have some of those hemoglobin dissociation curves, as well as a little bit of this regulation stuff that we're going to go through today before we move into the renal content. So I can see most of the class, or the majority of the class, finished that assignment on Wednesday that corresponded to the urinary anatomy. I did set the date for that to be due after break. So if you didn't, get to that by the time we circle back and keep thinking about renal stuff. In terms of what you should be preparing for, over the past years, there's been a revolving door of like A&P instructors. So I'm working to align our units to what Dr. Schlater is doing, since we're the two remaining now, which is to say that I'm going to move the acid-base stuff up to the beginning of the renal unit instead of being at the end as it was in your schedule. So I changed that because that way everything will work more smoothly for SI and stuff. And the person I was aligned with before left like 6 years ago at this point. So not all of the reading assignments are in those slides yet. I'm going to streamline them over break, but if you're looking for stuff, I made sure that you have the sections listed in that acid-base balance that correspond to what we'll be starting with. Some of that is in this regulation of ventilation chapter that we're doing now, and some of it jumps to the end of like chapter 19. So we'll start our renal unit with that, and then we'll proceed from there. So chapter 18 will in general come first, and then chapter 19, with the exception of this acid-base stuff. Okay, questions about that? Or like organizational questions? Okay, cool. All right, so I'll send out messages and hopefully it'll be clear what we're doing. But since you're here showing up, hopefully we're all on track. Okay, so today we're going to finish up with transport of gases, and we're going to rip through some of our regulation of ventilation stuff. Okay, so last time we were talking about, way last time, before our exams, we had talked a bit about the hemoglobin oxygen dissociation curve. So thinking about that, we thought about a couple of different effects. So one of them is going to affect this dissociation curve when we change altitude. So I want you to give it a go. Think about what kind of shift we'd see if we moved from New Orleans, which is actually a little bit below sea level, all the way to the top of Mount Evans, which is a very, very tall mountain. We're going to use this to remind ourselves what that dissociation curve is talking about as we move from talking about oxygen and how it's carried in the blood to talking about carbon dioxide. It's actually going to be a right shift. So what we were talking about with these dissociation curves, and I'll just draw what we're thinking here. We're usually thinking about right shifts is the kind of spoiler alert here. So what this dissociation curve is there's a relationship between our partial pressures of oxygen, which we know change between our systemic arteries and our systemic veins. Right, there's a drop in oxygen as we pass respiring tissue, metabolically active tissue. So there's a relationship between those partial pressures and how much oxygen is saturating our hemoglobin. We know that hemoglobin has 4 spots for oxygen, right? And in our veins, our quote unquote deoxygenated blood is actually about 75% full of oxygen, right? So it's not that there's none, it's just that there's less than in your arterial blood. Whereas our systemic arteries are at like 98.5% saturated. So pretty close to 100, but not quite there, right? So we can look at this difference, the unloading, as we refer to it, of that oxygen. So the difference between the amount of saturation in arteries and in veins, to think about how much oxygen went into tissue. And anytime we're in a situation where we really need extra oxygen, right? Like usually we're thinking your body's more active, but sometimes they're thinking low oxygen environments like we see in high altitude environments. We're gonna want to make it easier to get any oxygen to our body, to our tissue, to unload it. So when we express that in terms of this dissociation curve, right? What we see is something that moves to the right if we're making it easier. Because this means that if we're comparing the partial pressure in our veins and the partial pressure in our arteries, where they intersect with a curve that's moved to the right, gives us a way bigger amount of oxygen popping off and going into our tissue, which is really helpful if we're in a low oxygen environment, or if your body's using extra oxygen because you're making lots of ATP because you're doing stuff. And that's what kind of links together all the factors that shift this dissociation curve to the right. And anything that shifts us to the right, for example, increase in temperature, the opposite direction. So a decrease in temperature would shift us to the left. And we can think about that from a functional perspective too, right? So like if we're thinking temperature, skeletal muscle that's working hard producing ATP is producing heat, it's warm, we want oxygen there. So we want to unload oxygen. And to unload oxygen, that means we want a low affinity of hemoglobin for oxygen, because affinity is like how much they like each other. So if they're low affinity, that oxygen's kind of loosely bound, it's easier to pull it off, right? So we want that at the muscle. But for a left shift, that's something we might want like at the level of the lungs, right? You want to make it easy to grab oxygen from the air in the lungs, in the alveoli. So your lungs are relatively cooler than your skeletal muscle if you're going on a run, right? Because the atmosphere is a little cooler than it is inside your body. So that affinity for oxygen in that cooler environment in your lungs helps us grab and bind oxygen more tightly. So that's kind of the function of moving this curve to the left. Okay. So that's really, in a nutshell, all we're thinking about here when we're thinking about these many effects on hemoglobin and oxygen when we look at temperature, CO2 levels, pH, and oxygen levels using our altitude or 2-3 BPG as a kind of proxy for that. Okay. So that was what we were thinking about when we were thinking about oxygen transport and how we're putting oxygen into the hemoglobin of our red blood cells and moving them around. We are also moving carbon dioxide. It's really important to make sure that we're getting rid of carbon dioxide as we breathe out, because carbon dioxide, among other things, acts as an acid in your blood. And if you mess with the pH of your blood, you mess with a whole lot of stuff. So when we're thinking about carbon dioxide transport, a little bit of carbon dioxide travels on hemoglobin, not in those same 4 slots as the oxygen does, kind of over other parts of the hemoglobin usually. But most of the carbon dioxide transport is going to be happening directly dissolved in the blood. And when we talked about partial pressures and we talked about the solubility of different gases in blood, we saw that carbon dioxide is in general more soluble in liquid, more soluble in blood than the oxygen was. So that also kind of makes sense from that perspective, the different behavior of these gases. Okay. But in order to carry our carbon dioxide around, we're going to notice that we have the involvement of this enzyme called carbonic anhydrase, which is going to kind of convert the carbon dioxide into other compounds. So this is going to be important for what molecules we're actually looking for in the blood when we're trying to think about what's going on with breathing. So this is just a note showing you that there's more carbon dioxide in the plasma than there was oxygen. So we mentioned that before. And we can also see some of those other compounds that we're going to think about when we think about carbon dioxide. So some of it's going to be directly dissolved in the blood. Carbonic anhydrase is going to turn carbon dioxide into carbonic acid, which then dissociates into protons, which is really why we affect the pH with our carbon dioxide, and bicarbonate ions, which we'll talk about more as we talk about acid-base balance as well. So that's why bicarbonate is here on this chart about carbon dioxide. And here we can see a little bit of it bound to hemoglobin as well. So you can see most of it is actually going to be in this dissolved kind of converted form here. Okay. So here's what that reaction looks like between carbon dioxide and water using carbonic anhydrase as our enzyme. So we take our CO2 and water, which is everywhere in your body, inside your cells, outside your cells. Carbonic anhydrase combines those into our carbonic acid, which is what you see on the right-hand side of this equation. And the more carbon dioxide you have, the more this is going to happen and the more carbonic acid that you're going to create. From here, we would then be peeling off our hydrogen ions and leaving ourselves bicarbonate as well. Okay. And we'll talk more about that when we talk about the acid-base balance. So this will come back. Okay. So more products gives us more reactants. If we had lots of these, or sorry, more reactants give us more product. This is reversible. So if we had lots and lots of really we'll see in the future bicarbonate, this can run in the reverse. So while partial pressures of gases don't directly influence each other, because both carbon dioxide and oxygen are interacting with hemoglobin, there are some relationships between the transport of oxygen and the transport of carbon dioxide. So one we saw when we were looking at the dissociation curves, we saw we had the carbamino effect was one of the things that can cause a shift in that hemoglobin dissociation curve, changing the affinity of hemoglobin for oxygen. The reverse also happens. So the amount of oxygen you have in your blood does have a little bit of an effect on our ability to carry carbon dioxide as well. So what you're looking at here is that relationship. Okay, so for any partial pressure of carbon dioxide, if you have less carbon dioxide, there's less carbon dioxide in your blood. If you have more carbon dioxide, there's more carbon dioxide in your blood. That's pretty straightforward. And we don't have a like weird kind of integral shaped curve like we did for hemoglobin and oxygen. The other thing we can notice here is that we have like a family of curves, like we did in A&P1 for some of like our heart stuff. What's going on here is that if you have no oxygen at all in your system, you'd carry more carbon dioxide in the blood than you do in a fully oxygenated arterial or systemic artery, I guess I should say, under normal conditions. So there's a little less carbon dioxide kind of carrying capacity in these higher oxygen environments. So this is known as the Haldane effect. Okay. So here's a map of all of the kind of ways that we have oxygen and carbon dioxide partial pressures interacting with each other because of these effects primarily affecting hemoglobin. Okay. So looking here, you can kind of group these into three things happening. Right, so in low oxygen conditions, usually when we have low oxygen, especially because we're usually thinking low oxygen because you're using muscles producing lots of ATP. So low oxygen is usually going to correspond to high carbon dioxide. Right? Usually we have this kind of trade-off because our muscle is using oxygen, making carbon dioxide. So low oxygen usually directly creates more oxygen unloading, right? So usually in these low oxygen environments, we're going to make it easier to peel oxygen off our hemoglobin and put it into our tissue so that we can keep respiring, keep making ATP. So we do that directly. We have some ways we assist that process, right? That's kind of our goal, to make sure that our tissue keeps getting enough oxygen. So the Haldane effect was the one we just mentioned, right? So the Haldane effect in these low oxygen environments means we can carry more carbon dioxide, right? So here we see this higher amount of carbon dioxide. So that difference in that family of curves, that top one was with really low oxygen, okay? But when we have these higher amounts of carbon dioxide, the carbamino effect is going to come into play. So that presence of high amounts of carbon dioxide lowers hemoglobin's affinity for oxygen. Okay, so this lower affinity for oxygen means it's easier to peel off the oxygen. So we get more oxygen unloading. Okay. And at the same time, while we're in this respiring tissue, making lots of ATP, lots of carbon dioxide as a byproduct, carbon dioxide gets converted by carbonic anhydrase into carbonic acid, then dissociating into these protons and bicarbonate ions. And we also know that there's an effect of pH on hemoglobin's affinity for oxygen. So as we see the number of hydrogen ions going up, because we're basically converting this carbon dioxide, that's the Bohr effect. And it does the same thing as the others, increasing oxygen unloading by lowering the affinity of hemoglobin for oxygen. Questions about this process? So I recommend, for learning this stuff, is making a table and listing the effects and knowing whether we're making it easier to unload oxygen or harder, and whether we're raising hemoglobin's affinity for binding oxygen or lowering it. Okay. And this is just a figure showing you that the opposite kind of thing is happening in the lungs. So we have those same effects on there, but you'd see if you compare them one for one, we're looking at reversed arrows for all of them because we want to make it easier to load oxygen onto hemoglobin in the lungs. So we've got lots of oxygen carried around in our systemic arteries to the tissue. Okay. So now we're going to talk about the regulation of ventilation stuff. Okay. So when we're thinking about regulating ventilation, we want to think about nerves. So neural control of breathing by neurons is, we're thinking about nerves going to muscles. We want to think about the breathing rhythm being generated by the brainstem. And then we have some peripheral input to our respiratory centers as well. So like we have backflow of information coming back to the brainstem. So when we're thinking about our respiratory muscles, we know we're thinking about the diaphragm most importantly, but we're also thinking about external intercostals working with the diaphragm. And under certain conditions, so when we're forcing our breaths, we're also thinking about internal intercostals and abdominal muscles being involved in pushing out extra hard breaths. Okay, so these are all named muscles, which tells us that they're skeletal muscles. They're attached to your skeleton, which means they're part of your somatic nervous system and they're controlled by motor neurons. Okay, so we talked about those somatic motor neurons in the past. In A&P 1, we talked about acetylcholine. We looked at that whole relationship, how that trigger works. Okay, now we're just going to name some nerves. So you've probably seen the phrenic nerve in the lab. So for breathing in, that's the responsibility of our diaphragm and our external intercostals. phrenic nerve is super important, goes to the diaphragm. It actually comes from your cervical vertebrae from C3, 4, and 5, keeping that diaphragm alive. So that's inspiration. And for our intercostal muscles, the nerve is just named after the muscle. So our external intercostal nerve would go to the external intercostal muscles. So for expiration, usually passive, usually just the diaphragm relaxing, but active expiration, we've got those internal intercostals activated by that internal intercostal nerve. So here is what action potentials in those nerves would look like tied up with our breath cycle. So you can see, let's focus here, right? So you can see this curve up and down. Looks an awful lot like that little curve we saw for our tidal volume for our tidal breaths, right? So that might kind of trigger that. And we can see, yeah, those tidal breaths here in our lung volume as well. So we can see that our phrenic nerve and our external intercostal nerves are activating during that contraction phase as we're breathing in. And your internal intercostals and expiration under passive, quiet breathing are doing absolutely nothing, right? Because we don't need them for just quiet tidal breaths. But if we want to take big, deep breaths, we're going to have to breathe out extra hard. So that's when we see those internal intercostal nerves kicking in, as well as the tension in those muscles. We can see now we actually do have some tension generation there. Cool. All right. So those nerves are part of your peripheral nervous system because they're out in the periphery. Okay. We also have parts of our central nervous system involved that are kind of controlling breathing rhythm. So we do have a number of slides about this central nervous system. I'm going to talk through it for your interest. I'm not going to test you on the stuff about the pons and medulla that I'm about to say to you, so I'm going to go a little fast, and I'll let you know when we're coming back to stuff that's going to be on the exam. Okay. So when we're thinking about what's going on in the brainstem, you learn the main parts of the brain, the parts of the brainstem. This is some of the important stuff that they're doing. So lots of really important critical for life homeostasis functions are happening in your brainstem, like breathing. Okay. So we have inspiratory and expiratory neurons in your brainstem in respiratory centers. So you'd have like little clusters of neurons in these areas that are responsible for controlling breathing in, inspiration, breathing out, expiration. And we have some neurons that do both a little bit. So here's what we know, a little bit about where they are. So here are those parts of the brainstem you hopefully remember from lab. We had the medulla oblongata, we have the pons, and we have the midbrain. So it's the pons and the medulla that are really responsible for breathing rhythms. So here you can see where you might find those neurons related to inspiration and expiration. They're kind of color-coded here. So our expiratory neurons are colored in yellow. Our inspiratory neurons are colored in blue in this figure. And we can see that we have a more ventral group of neurons. Ventral means like towards the belly. Think about your, I don't know, like your vents for your body are on the front side. So we got a group of them there. And then we have a dorsal group. Dorsal, think dorsal fin of a shark on your back. There's also a dorsal group here related to these. And we know that they're responsible for controlling breathing in part because we can pair up their membrane potentials and their action potentials to actual moments where you're inspiring or expiring. And they do this cool kind of ramping up thing as you're breathing in. And we know expiration is usually a passive process, so it's not too surprising that they just turn off during expiration. We also have some in the pons. So in the pons, you can see the pontine respiratory group up here in green. Okay. This is also sometimes referred to as your pneumotaxic center, and it has all the different types of neurons as well. We don't know exactly how it works or exactly mechanistically what it's doing, but we think it's maybe regulating how you move from that moment where you're breathing in. We know we're coming to equilibrium with the environment as we hold the most air. And then when you switch to breathing out, we think it is maybe doing something related to that. There's also a central pattern generator, establishing the whole cycle and whole rhythm. We think it's somewhere in the medulla, but the exact location and the exact mechanism of how it works are unknown. So we've got lots of cool kind of neuroscience questions open for the future, at least as of the date that your textbook was written. I did a very quick search to see if they know exactly how it works now, and not yet, but I didn't look that deep. So we may know more soon. Okay. So that's what we are thinking about related to that. Okay. So when we think about what's controlling the rhythm, right? So our control of the rhythm going out to the body comes from here, right? So this is where we would be signaling out to our muscles, right? Like the diaphragm, like the external intercostals. So we know that there is some involvement of the inspiratory and expiratory neurons, primarily in the medulla, but maybe a little bit in the pons as well, okay? We also have the ability to think about breathing, right? You can force yourself to breathe in and out to different depths. You can force yourself to breathe like very shallowly or very deeply. So your cerebral cortex would do the voluntary part of that if you're actively thinking about it. But we also have kind of automatic reactions to sensory input, right? So we'd have signals coming in from your lungs, and as we're going to find out, coming in from various receptors like attached to your cardiovascular system that are going to tell your brainstem, okay, something's going on and it's important for us to change our breathing pattern now. So some of these, the ones we're actually going to focus on, which is where testable information will actually pick up again, will be our chemoreceptors. So central and peripheral chemoreceptors are going to be directly sensing changes to partial pressures of the gases that we're interested in, so carbon dioxide and oxygen. But there are other things that would also affect when it's a good time to breathe more deeply or more frequently. So your body is able to sense how inflated your lungs are. In humans, this doesn't make a huge difference, but if you are interested in becoming a vet, it does in some other species. So some species really react to like overinflation of lungs. We certainly do react to these irritant receptors. So if you inhale like a bunch of smoke, that would affect these irritant receptors. And there are some gases like sulfur dioxide that would also physically irritate your lungs and therefore change your breathing patterns. We also have some proprioceptors that are giving information here too. Proprioception is like sensing the position of your body. So your proprioceptors can sense that like your intercostal muscles are moving, stuff like that. So sensing your own position of your body and movement. Okay. So these are all peripheral inputs. So inputs from out in the body, things we're sensing that are giving information to our respiratory centers so that we'll alter the breathing rhythm accordingly. Okay. So we're going to focus specifically on the chemoreceptors. So here's where we're back to stuff you want to thoroughly understand. Okay. And back to stuff science more thoroughly understands. So our chemoreceptors that we're interested in are going to be detecting blood levels of both oxygen and carbon dioxide, although we're going to find they're not responsive to them equally. So we'll take a look. So chemoreceptors, the chemical that we're thinking about, are these nonpolar gases that we're transporting around the blood. So we've got them in two places. So we have some of them in the carotid bodies. And those are really the peripheral ones because they're not in the periphery. So your carotid body is going to be located like in your carotid sinus. So your carotid arteries come up your neck and then they split into internal and external. That carotid sinus is a swelling right near the split. That's where you would find the carotid body and these peripheral chemoreceptors. We also have chemoreceptors directly in the medulla oblongata, so directly next to the blood in the brainstem. Those would be central chemoreceptors. And they behave a little bit differently, which we'll walk through. Okay, so here we see the peripheral ones. So this is what I'm talking about regarding their location. So here we see carotid arteries coming off the arch of the aorta. We got a common carotid for a while. Here you can see that carotid sinus, that swelling, just like your sinuses in your nose are like big hollow areas of your bone. This is a larger hollow area in the carotid. So here you can see that we have a bunch of sensors here. So your carotid bodies are going to be here. We also had baroreceptors here in the carotid sinus. So we mentioned before that this is also a place for sensing like pressure of the arteries. Okay. So here's the bifurcation of the carotid as we go into internal and external, up the neck and up to the brain. Okay. So that's where you would find them. So they're in direct contact with this fully oxygenated arterial blood, that's systemic arterial blood coming out of the left side of the heart, right? We're going here like first, basically. So these peripheral chemoreceptors are going to be communicating using afferent neurons. So there's going to be chemical messengers involved here in order to send signals to the medulla oblongata, to those respiratory control centers that we mentioned. Okay. So when we think about what they're sensing, they're mainly going to be reacting to changes in carbon dioxide, which might be a little surprising. So I try to keep repeating it because it was surprising to me when I learned it, that for your body, rising levels of carbon dioxide buildup is more of an immediate problem than lowering levels of oxygen. So very much too little oxygen is going to be a problem, but you have a much wider range of oxygen that is acceptable to your body than when we're looking at carbon dioxide. So we're going to see that these peripheral chemoreceptors are mostly responding to carbon dioxide changes, partially through that conversion of carbon dioxide into hydrogen ions and bicarbonate ions that's affecting the pH. Okay. So what we're looking at here is how we see changes between, in this case, partial pressure of oxygen, so how much oxygen you have in your blood, versus the minute ventilation. So the minute ventilation, we know that we calculate that by calculating our tidal volume times our respiration rate. So what this really is how much air total are you taking into the lungs per minute? Okay. So we can see that our normal partial pressure of oxygen here gives us a certain minute ventilation, right? As our partial pressure of oxygen drops, it can drop kind of a ways before we start to see major changes in the minute ventilation, all right? So it's not till we get to like, I don't know, let's say like 60, before we start to see this really kick off and start to change your breathing, largely by changing your breathing rate at these lower levels of oxygen. However, if we look at what's going on with carbon dioxide, we see immediate changes. Right? So normally your systemic arteries have a partial pressure of carbon dioxide of 40, right? In the veins, that would jump up to 46, corresponding to a relatively low, easy breathing, normal minute ventilation. But you can see here, as soon as we start to have carbon dioxide buildup, like any at all, that minute ventilation immediately starts to rise. So as soon as we start to see rises in carbon dioxide in your systemic arteries, you are immediately, hopefully, going to start breathing more quickly and more deeply in order to breathe off that excess carbon dioxide. This is because if you get up to 90 millimeters of mercury of carbon dioxide, you're going to go into a coma. And if you get above 180, you are going to die. So this is that immediate problem of changes to carbon dioxide levels, right? So we can see that once we get up this far, right, big problem, right? Versus in our arterial partial pressure of oxygen, right? It wasn't until we changed this by like 40 or so that we start to change our breathing significantly, versus if we change our PCO2 here by 40, we're about to go into a coma, right? That is not an acceptable amount of time to wait to change your breathing in order to deal with this problem. Okay. So our chemoreceptors are sensing these changes, and they're particularly responding as soon as we see changes in carbon dioxide concentration, partially when we're thinking about directly what would respond to the receptors by responding to pH changes, because carbon dioxide ultimately acts as an acid in the body. Okay. This is particularly true for the central chemoreceptors. So the central chemoreceptors are our second place we have chemoreceptors. The peripheral ones are in the carotid sinus. Central ones are on the ventral surface of the medulla, so like the front part of the medulla. And they're responding to pH changes in the CSF that's neighboring them. Okay. So your peripheral chemoreceptors do somewhat respond directly to carbon dioxide, but when we're looking at the brainstem, we know there's a blood-brain barrier. We know the brain is kind of protected from a lot of things. So it is protected in part from some of these carbon dioxide changes, but it is still going to be able to sense the pH changes that carbon dioxide is causing in your body. So we'll take a look at that in a figure in a second. But a good thing to know about these central chemoreceptors is that these ones just don't respond to oxygen changes at all. So they are purely focused on assessing these carbon dioxide changes by assessing pH. They're not responding to oxygen. Only the peripheral ones would be responding to oxygen, and only when we have really big changes. Okay. So when I said big changes, what value of PO2 would that be? Yeah, about 60 is when we start to see major changes. Okay, so here's a picture of what's going on with the central chemoreceptors. So what we're focusing on here is that central chemoreceptors are going to react when we have a change in carbon dioxide in the blood. So here we see that carbon dioxide in the blood, but they're not touching that bloodstream directly. We have a blood-brain barrier. So what they would be directly sensing is the fact that this carbon dioxide mixing together with water is getting converted by carbonic anhydrase into carbonic acid, which dissociates into bicarbonate ions and hydrogen ions. And these hydrogen ions, pH, that H is actually for hydrogen ions, right? So it's that H, that hydrogen ion, that is getting directly sensed by our chemoreceptors, the central ones. That's what we're focusing on there. Okay. So here is what kind of flow chart we would see if we were just walking through our response of these things to each other. So you can see that our peripheral chemoreceptors are detecting and responding to all kinds of signals, right? So directly responding to carbon dioxide. They're also sensing those pH changes. They also sense major changes in oxygen levels, major drops. But our cerebrospinal fluid and those central chemoreceptors are only able to respond after we convert those high levels of CO2 into pH changes. But overall, any of these signals is going to change ventilation, right? So any way that we sense rises in carbon dioxide is going to make us breathe more often. If we have a real noticeable drop in oxygen, hopefully our peripheral chemoreceptors kick in, also increasing our ventilation in those circumstances. So ultimately, what would have caused these changes in carbon dioxide, in pH, also drops in oxygen, might be something like usually hypoventilation. So hypoventilation was not breathing enough, right? So if you're breathing too slowly, means you're not breathing out carbon dioxide, so it's building up in the blood, right? So that's why we might have it there to sense through our peripheral and also central chemoreceptors if we're thinking about the pH part, leading to increases in ventilation, hopefully putting us back to normal. Cool. All right. So our final topic here when we're thinking about regulation is we have a relationship between how much we're breathing, so the ventilation piece, and how much blood flow we have in the lungs passing by those alveoli. So this is reflected by the ventilation perfusion ratio. And we're going to try to match our ventilation and our perfusion. So we're going to try to match how much air we're taking in and how much blood is passing by that air in order to do the most effective breathing we can. So this is going to be under local control. So both the breathing part, the ventilation part, and the perfusion part, the blood piece, are going to be responding to local signals. Okay. So we have a ratio here, our ventilation perfusion ratio, which is literally just a ratio of ventilation on top. So rate of airflow is on top. Rate of blood flow, do not know why they used a Q for this, but rate of blood flow is on the bottom. So ventilation over top, perfusion below. Okay. So we want to regulate these so that they match each other, so that we are effectively breathing in air and doing our gas exchange. So we need both fresh air and we need like fresh blood to carry those gases. Okay. So here's what this looks like. Right. So here's our ventilation, our air coming in. Here's our Q, our perfusion, that rate of blood flow going past, right? So we know that under normal conditions, right, we are coming to equilibrium and we're creating a partial pressure in both the alveolus and in this blood of 100 millimeters of mercury if we're looking at partial pressure of oxygen, 40 if we're looking at carbon dioxide, right? So this is the result when we effectively match that airflow to the blood flow. So usually we want to see a ventilation perfusion ratio of 1. So matching airflow and blood flow is going to give us 1. Okay. Now, sometimes we're going to have problems in the body, right? So one problem we might see would be if an airway gets obstructed. So like you're choking, right? And you're not getting air, right? This is going to create a decrease in ventilation, right? So you're not breathing as much. So this ventilation number is going down. But this doesn't immediately change anything about the blood flow, right? Your blood doesn't know yet that you aren't actually getting fresh air into that alveolus. So when this first happens, you're not going to see any change in Q, which is our perfusion, our blood. So we're going to see this ratio dropping below 1, right? So our ventilation is now going to be less than our perfusion. So the result of this immediately is going to be that our partial pressure of oxygen drops because we didn't get fresh air. And our partial pressure of carbon dioxide is going up because we didn't breathe out that carbon dioxide. And so now we know this is going to start creating a problem because we're starting to see this buildup of carbon dioxide in the blood here. Okay. So below 1 is a problem, and this indicates that we have an obstruction. This is a ventilation problem. We can also have an obstruction from the other side. So say you have a blood clot. So you've got like a heart attack, stroke situation, except we're affecting the lungs here. So a pulmonary embolism, say. Okay, so in this case, you'd still be breathing, you'd still be getting air to the alveoli, right? So you would still have a normal number for your ventilation, right? But now we see that Q is changing. So Q is our perfusion, Q is our blood, right? So now we're dividing by a smaller number. So we're going to see that ventilation perfusion ratio go up. So now this is going to be above 1. Above 1 versus below 1. Let's do that. Okay, cool. So the results of this are going to be that we see increasing levels of oxygen in the blood and decreasing levels of carbon dioxide after we have that obstruction of the blood vessel. And we're going to want to adjust our rates here as well. Okay. So we'll kick off here next time, but these are the reactions that we're going to see in response to those two situations. So when we talked about arterioles, that we can vasoconstrict and vasodilate. When we talked about bronchi, that we can bronchodilate or bronchoconstrict. Here's where they come together. Okay. We will stop there.